molecular biology.
That's true. That's true. Yeah.
Yes, sir. We are on live now. In 5 minutes, we will be just starting it.
Okay.
Meanwhile, the audience are joining.
Molecular biology.
That's true. That's true.
Yes, sir. We are on live now. In 5 minutes, we will be just starting.
Okay.
Meanwhile, the audience are joining.
Can you share that YouTube link with me, Alamuru? So if somebody is asking, I can share this thing with him.
Definitely, sir. In 1 minute, I will be sharing.
Sure.
Should I keep in WhatsApp, sir?
Yeah, you can do in WhatsApp or even in this Zoom chat, wherever you would like to.
Okay.
Got it. Thank you.
Hope we can start now, I think. Yes, Pratishtha. Yeah.
Yes.
Yes.
Good morning, everyone. Distinguished participants, ladies and gentlemen, I am pleased to welcome you to the webinar on genome editing
Yes,
to
Pratishtha.
enhance multiple disease resistance in crop plants. In this virtual conference room, I see such amazing guests and dignitaries from around the world sharing same interests and enlightening one another with the advancings in the field of agriculture. I'm sure we are also reaching out to many new practitioners, technologists, innovators, business professionals, and civil society representatives, fellow mates, and many more people. A warm welcome to you all. We all understand the importance of science, technology, and innovation in our day to day lives. and in the ways in which they are transforming the world. If this was not clear earlier, it is blindingly obvious now, alongside the intricacies arising with the hour at hand. Science, technology, and innovation are indispensable for our response to and upliftment from the current and the forthcoming scenarios. Equally important are deployment of scientific inventions, innovations, and technological solutions on a scale that will reach the target audience. So, why here? Why today? And what is the purpose? We're all embraced by the presence of the distinguished and well rounded dignitaries to shed some light upon the varied measures to be taken under consideration for genome editing in order to attain enhancements for disease resiliency in plants. All our online webinars aim to discuss leading edge technologies and recent scientific developments in addition to the immediate challenges in agriculture and allied sectors. The idea is to familiarize students and professionals about current research trends, relevant and pioneering technologies in plant genomics for high level analysis, and interdisciplinary areas that are embraced in leading laboratories by the excelling scientists. Plantgenomia is a non-profit organization promoting and publicizing genome sciences and investigations in the field of agriculture across the globe. We organize various webinars and developments comprehending genome research in plants for all students, researchers, industrialists, and professionals internationally. Here at Plantgenomia, we hold a global team of reputed scientists and research scholars from various parts of the world, distinct states of India, the United States of America, United Kingdom, Nigeria, Philippines, and Spain, etc. Technical support is laid to us by the scholars from the Department of Genetics and Plant Breeding. Our motto is to disseminate every bit of knowledge among all. We believe that no research is accomplished until broadcasted, and that is an ongoing process through layers and lapses of time. I would also like to extend my heartiest welcome to the senior dignitaries, respected chief guest, Dr. Rajiv Varshney from International Crops Research Institute for the Semi-Arid Tropics of India. Our speaker today, Dr. A.C. Kushalappa from McGill University, Canada. Our media partner, Just Agriculture Magazine, the core subject specialist team, the core support team, our esteemed founder, Mr. Alamuru Krishna Chaitanya, and the lovely audience. Now, I would like to invite Dr. Rupali from the University of Western Australia to introduce our revered chief guest, Dr. Rajiv Varshney.
Thank you, Pratishta. And thank you everyone involved in Plantgenomia for giving me the opportunity to introduce our honorable chief guest for the event, Dr. Rajiv Varshney. Rajiv is the Global Research Program Director for Genetic Gains at ICRISAT, which is International Crops Research Institute for Semi-Arid Tropics. He has a PhD in agricultural botany from Chaudhry Charan Singh University, India, and has worked at IPK Gatersleben, Germany for 5 years before joining ICRISAT. He is internationally renowned for his work in the area of plant genomics, genomics-assisted breeding, translational genomics, and capacity building in international agriculture. He joined ICRISAT in 2005 and simultaneously served as leadership for consultative group on International Agricultural Research Generation Challenge program hosted by CIMMYT, Mexico. At ICRISAT, he has led teams that have sequenced some of the most important dryland crops, including pigeon pea, chickpea, ground pea, groundnut, and pearl millet. He has provided strategic leadership to Tropical Legumes Project that has facilitated the release of 266 improved legumes varieties with a benefit to over 25 million smallholder farmers in 15 countries in sub-Saharan Africa and South Asia, which is a massive number. So Dr. Rajiv is a recipient of most coveted Shanti Swarup Bhatnagar Prize, which was honored by Honorable Prime Minister Shri Narendra Modi ji. Dr. Rajiv's outstanding contribution to agricultural research was recognized, and he was made a recipient of Indian Council of Agricultural Research Rafi Ahmed Kidwai Award. He is also a recipient of several prestigious awards, including Professor Jayashankar Lifetime Achievement Award, G.D. Birla Award for Scientific Research, Professor Lalji Singh Achievement Award, and the list goes on. Dr. Rajiv is a prolific, highly prolific indeed, author listed among highly cited researchers for 7 consecutive years. He has published more than 450 high-quality research articles, including 15 papers in Nature Journal. He is listed 123rd position in world ranking in plant biology and botany category in the study from Stanford. He holds global collaboration and holds adjunct position with more than 10 organizations. He chairs several committee editorial boards funding organizations and advisory board. Dr. Rajiv, thank you for accepting our request to be a chief guest for our international webinar on genome editing in plants. It is our extreme pleasure to have you today, sir. I now request you to speak a few words about your research in genomics. Thank you again, and over to you, Dr. Rajiv.
Thank you very much, Rupali. It seems you have really done extensive research on my biodata. So I'm really humbled, and I feel honored and humbled to listen these kind words. Anyway, good morning,
Thank you very much.
good afternoon, and good evening, wherever you are. And I'm really very excited to be the part of this event. And let me thank Plantgenomia and especially Alumuru and all these other colleagues for giving me this opportunity. I should mention that you guys are doing a brilliant job and basically promoting the genome science among the students, community, scientific researchers around the world. So you are doing really marvelous job. Congratulations to all of you, and many thanks for all your efforts. And as I mentioned, I'm really thankful to all of you for giving me this opportunity. And I would like to welcome you all who are our audience. I think that this number is continuously growing, and I think more than 5,000 people, they registered for this webinar. So this is really exciting. And I'm sure that this will be available as a YouTube link. So maybe the people who are not able to join now, they can listen this thing at a later stage as well. So anyway, and I'm really very happy that this webinar is really on a very, very topical subject. And more than that, we are having really internationally renowned scientists as a speaker for this webinar. And I will talk about speaker a bit later, but I think this is really exciting that we are talking about genome editing technology. And Dr. Kushalappa, who is the professor at McGill University, he's expert in this particular technology in area and in context of plant pathology. So before we start seminar, let me give a brief background that why we are sitting here. And I think that other speakers and basically Pratishtha, he already gave a little bit background, but let me talk very briefly. That we all know that agricultural science community is having a duty and obligation, in my opinion, to contribute towards feeding an ever-growing population. And it is interesting to note that it took about 200,000 years of human history for the world's population to reach 1 billion. On the other hand, it took only 200 years to reach about 7 billion people. So what does it indicate? That population is growing exponentially, and we expect this number to reach around 9 billion by 2050. Furthermore, we all know that we are having a global issue of climate change, and this will not have only implications on abiotic stresses. Many times we think about the heat and drought, but also may bring some new diseases about which we are not aware right now. We all are aware with the current COVID-19 pandemic. Until this started, even in the late 2019, we never knew that this is going to happen, and this will have the implication that the whole world will be shutting down. So we all are aware that what kind of challenges we may have, the society may have. And not only in the future, that agriculture, especially in the developing country, that they will be facing a number of challenges in the area of biotic and abiotic stresses that will be hampering the crop productivity. I think this is the issue right now as well. So to address these issues at present and to keep ourselves prepared for future, we need to work towards enhancing crop productivity and developing climate change ready crops. In this context, traditional breeding technologies have been playing very important role, and majority of the varieties which are feeding the world, they are coming from the technology. So thanks to all of them. But now to match the current rate or the future requirement, it is really essential that we need to embrace new technologies for crop improvement. We all are aware the success stories of a variety of breeding technologies, including hybrids, mutation breeding, and in recent years, genomics assisted breeding, about which Rupali was talking, some of our work as well, that we have decoded genome sequences, and more than that, we have translated the genome information in developing the new varieties. And we also have seen the success stories of genetic engineering technology. In the same series, we are having now a new breeding methodology that's called genome editing or gene editing. In fact, we all know that 2020 Nobel Prize in Chemistry was given to Emmanuel Charpentier and Jennifer Doudna for their discovery and development of CRISPR-Cas9 genome editing. So this highlights the role of this important technology. And nowadays, this technology is already being extensively used in agriculture sciences. I will not talk much about this technology because we have the expert in this area. I have not having any expertise other than in this area other than reading, but nevertheless would like to mention that it's really important that for gene editing technology, we should have the gene, not the candidate genes, I'm talking the gene, the causal gene for a trait, which we would like to improve. And good news is that due to advances in genomic science and systems biology, it has been now possible to identify such causal gene, though it's challenging, but possible. So I think that this is right now very exciting time that with the help of the genome science, with the help of the multidisciplinary science and technologies, we can really isolate, we can have the gene for manipulation through the different technology. So that's what I would like to mention at this stage, Rupali, and will be contributing further. Thank you very much once again for giving me this opportunity.
Yes, Pratishtha. Pratishtha.
Yes. Much gratitude to you, sir, for appraising us all with such knowledge and insight, for accepting our invitation and being a part of this event. It's been a stupendous commencement indeed. Next, we have the much admired founder of Plantgenomia, Mr. Alamuru Krishna Chaitanya, to address the audiences and guests present with us today and to mark the initiative taken up by Plantgenomia and his team.
Thank you, Pratishtha. Dear distinguished chief guest, Dr. Rajiv Varshney from ICRISAT, esteemed today's speaker, Dr. A.C. Kushalappa from McGill University, reverenced media partner, Just Agriculture, DPS Badwal, scientists, professors, industrialists, senior executives, and my colleagues. A very warm good morning to everyone. First and foremost, I would like to express my heartfelt thanks to all for taking time to attend this international webinar on genome editing to enhance multiple disease resistance in crop plants organized by Plantgenomia. The way moving forward, together with us, we have distinguished speakers who not only command a high authority in sharing their views, but also a living legacy in scientific research. Our speakers will share with us on the valuable insights on genome editing to increase multiple resistance in crop plants. Day by day, we are having a lot of advanced scientific research which portrays how much potential the scientists are working in. From conventional to modern CRISPR technology, genome editing was a marvelous phenomenon which can revolutionize the future agriculture. The planet, as we know, continues to get smaller, complicated, and literally flat. But we see now that being connected is insufficient. At this very moment, research, talks, discussions, and even execution by global leaders, top researchers, and prominent CEOs have already underway that holds a new potential. The planet is becoming smarter, thus facilitating the movement of everything from agriculture to science, and that helps billions of people to work, eat, drink, sleep, and improving the quality of living. It is in full awareness of a smarter planet. Plantgenomia has taken an initiative in meeting with all the world-class scientists to create a smarter dissemination virtual system by making the use of the knowledge and advanced technologies which benefits all the researchers to get maximum results. I was very happy to see the overwhelming response from participants from many countries touching almost every part of the globe. Once again, on behalf of Plantgenomia, I like to sincerely convey my thanks to all in making this event possible. I do hope that you'll find the meeting meaningful and memorable. Thank you, one and all.
Thank you very much, Mr. Krishna. Those are some very gripping words coming from you. While we are presenting a gesture of gratitude, I would also like to thank Dr. K.P. Vishwanathan, Vice Chancellor of Mahatma Phule Krishi Vidyapeeth, Rahuri, and Dr. K.V. Peter, former Vice Chancellor of Kerala Agriculture University, for giving their valuable forward message on Plantgenomia webinar. Moving further, we would like to express our indebtedness to our chief guest, Dr. Rajiv Varshney, for his inestimable presence. And now, as we subsequently proceed with the events, we would like to express our heartiest pleasure on having you here with us to chair the session and introduce our today's speaker, Dr. A.C. Kushalappa.
Thank you very much, Pratishtha. And thanks once again, Alamuru, for very nice background. And now we would like to introduce you, Dr. Azmat Kushalappa. And as I mentioned earlier, once again, we are very much excited to have him. And I think that it is really an honor for all of us to listen his talk on this important and exciting topic. He is a professor at the McGill University, Quebec in Canada. He is a well-known scientist in the area of plant pathology and crop biotechnology, has published more than 100 papers in international journals. He has been recognized for his outstanding work by many organizations, but I would like to mention one of the very important award. He is recipient of the Dr. and Mrs. D.L. Wiley Award from the Canadian Phytopathological Society for an exception and distinguished contribution to plant pathology. He has been an invited speaker on plant biotic stress resistant genes and genome editing at 16 national and international conferences. Wow. In the past 6 years. He is a member of the National Science and Engineering Research Council of Canada. This is called NSERC funding panel. And also he is serving as BMC Plant Biotechnology as the associate editor. He has trained more than 50 graduate students and postdoctoral fellows, and now many of them, they are serving the different institutes around the world. His current research focus is the identification of plant biotic stress resistant genes through forward and reverse genetics approaches, including systems biology, and then the use of these genes to replace the susceptible genes in commercial cultivars based on the cisgenic engineering, where you are transferring the gene from the sexually compatible plants, as well as by using the CRISPR-Cas9 systems, about which he will be talking now. So with an overall objective to enhance the genetic diversity and crop improvement. So we are very much honored to have you, Dr. Kushalappa, and we know that this is very late for you, but you have kindly agreed to deliver this talk, and we really appreciate your commitment on your contribution to the Plantgenomia. So once again, I would like to request all of my colleagues from here, Plantgenomia and audience, to welcome Dr. Kushalappa for delivering his talk. So Dr. Kushalappa, floor is yours. Thank you very much once again, and we are very excited to have you. Floor is yours, Dr. Kushalappa.
Thank you very much, Dr. Varshney, and good morning to everyone, or good afternoon, wherever you are. And I thank Dr. Varshney for a comprehensive introduction. And also, I would like to take this opportunity to thank Mr. Chaitanya and his team for inviting me. And they are the youngest scientist ever invited me in my whole career. Okay, keep it up, young boys, girls, keep it up. Okay, so with that, can you all see my slides?
Yes, we can. Thank you.
Okay, so today I would like to speak on genome editing to enhance multiple disease resistance in plants. I'm not going to give general introduction to the significance of plant diseases. Dr. Rajiv Varshney already mentioned few important things. Okay. Before we get into these, what is genome editing based on CRISPR-Cas9? This is just a tool or technology. So in order to genome edit genes, what are these genes? We have to have a in-depth knowledge on these science behind these genes and plant pathology. And this is what I'm going to cover today. So it will be rather basics to see how we can select genes to enhance disease resistance. First, let us see where these genes are localized. Plants have trillions of cells. In each cell, they have nucleus, and the nucleus contains chromosomes, and they contain genes. And these genes are double-stranded helix. And these cross walls are nothing but base pairs or ATGC. And these genes determine what the phenotype is going to be. Okay. Now, if these genes are so important, where did they come from? What is the origin of these genes? 13 billion years ago, there was a big bang, and the cosmic rays joined together and produced the proton and neutrons, and they were enclosed in a nucleus, and electrons orbit around this. This is atom. And several atoms joined together and produced the planets about 4.5 billion years ago. And also later on, these atoms joined together and produced genes, and they produced cells. And these are the single cells, and which later on gave rise to complex uh, cellular complex cells. Now, how did the atoms evolve into genes? Atoms first evolved into organic molecules. For example, in nature, two hydrogen and one oxygen makes water, similarly methane and ammonia. In lab, some scientists put all these things and subjected them to high electric spark, and they were able to generate small organic molecules. These things happen in nature. So now these compounds can combine to produce RNA. This RNA will lead to the production of DNA. And these RNA and DNA lead to the production of viruses. Well, I'm not going to cover details on virus because they have so many other theories. And this DNA evolves into genes, and the genes evolve into cells. Now, a few atoms, not all the atoms known so far, but only the few atoms evolved into genes. This is the double helix, and these helix consist of pentose sugars, either ribose or deoxyribose. These sugars are connected with phosphoric acid, and also they are connected to nucleotides. These are the ATGC that we all know that is being sequenced. And 3 nucleotides code for an amino acid. And several amino acids combined together produce proteins. So now, how the genes function? Okay. Now the DNA transcribes into RNA, RNA translates into protein and enzymes. The enzymes are catalysts. And the plant biosynthesizes glucose in Calvin cycle, and then the glucose is biosynthesized into several metabolites, more than 200 metabolites by these enzymes. Now, the proteins and metabolites are used in plant growth and function. Now, the genes evolved into different organisms. Okay. For example, we have mainly prokaryotic cells and eukaryotic cells. And the prokaryotic cells do not have nuclear membrane. They don't have nucleus, but they have the chromosome. And in that chromosome, several genes are localized. These single cells gave rise to several single-celled organisms such as archaea and bacteria. And the genes in these cells evolved into membrane-based, organelle-based cells. There are several organelles with membrane. They are very complex, and they also contain several chromosomes, and each chromosome contains several genes. These complex cells led to the evolution of multicellular organisms such as plants, animals, and fungi. Now, the microorganisms slowly evolved into pathogens. They were saprophytes, meaning they live on dead organic matter. They may be endophytes. They live inside the plant but not pathogenic. They may be biotrophs. They are pathogens requiring living hosts to multiply, like rusts and powdery mildews. And necrotrophs, which produce enzymes and toxins and kill the host and then feed on them, like botrytis and Fusarium, etc. And there are hemibiotrophs. They start as biotrophs and then they become necrotrophs. Typical example is Phytophthora. However, several of these necrotrophs also are considered to have a short biotrophic phase. And these things are also very important in understanding the biology of the pathogen and how they enter into the plant. And we can use this biology to kill the pathogen at its weakest point. Now, evolution of plants. For more than 10,000 years, we have been domesticating plants. And the nomadic people, they were domesticating not only animals, but also plants. And they selected the best cultivars, and they, with the high yield mainly, and also they may have selected some with disease resistance. Those that were susceptible may have dead and gone. So now, breeding has been used to improve plants. And though it is occurring in nature, artificially, people bred several crossed different plants with different traits. And they were able to develop dwarf plants with no lodging, which facilitated mechanical harvest. And also they selected plants that respond to high doses of fertilizers and also give very high yield. The high yield and the dwarf plants led to industrial revolution. These led to large farms, and these large farms increased diseases and pests. And to combat these things, researchers invented pesticides. And everywhere pesticides were applied, and it led to environmental degradation. To increase the yield, they applied a lot of fertilizers, which led to the degradation of soil. Now, if you look at nature, here is our plant, and it absorbs organic matter and minerals from soil, then takes carbon dioxide from atmosphere, and it biosynthesizes carbohydrate and proteins and also releases oxygen. These are taken up by plants, animals, and microbes. When they die, they decompose, become organic matter again. This is how the atom is recycled. Now in nature, what happened? There is a perfect balance. And the green revolution has led to a lot of pollution in the agro ecosystem. And this has to be reduced. Otherwise, we may not have good air to breathe and food to eat. Now, to mitigate this, the marker-assisted breedings were developed. They developed new cultivars. Molecular markers are nothing but segments of chromosomes linked to traits. And success of molecular assisted breeding is they could pool multiple traits, especially agronomic traits and also yield mainly. And they developed more than 200 cultivars for each crop. So now they had some limitations too. It's a breeder's nightmare to combine all the traits that a commercial producer wants. Often they look for yield and agronomic traits, but the disease resistance was poor. It was not acceptable to the public demand. And also it takes more than 10 years to breed a cultivar. And markers or quantitative trait loci are often not associated with traits. The complex traits are controlled by hierarchies of genes, not one or two or three, and they are not located in a small segment of a gene. Now, genome editing to improve traits. Okay, this is the next step. Okay, this can be used to improve several traits, but today I'm going to focus only on enhancement of multiple disease resistance. Genome editing is a precise modification of genes. In marker-assisted breeding has produced hundreds of cultivars, or at least 200 cultivars for each crop. So now these, for sustainable crop production, the plant must have, the nature should have genetic diversity or the crop diversity. But also, to grow a plant, we have to have a genetic composition, okay, for a given trait. So in addition to crop diversity, we also have to increase genetic composition of for a given trait. And that, I think, we can do it by genome editing. Now, what is the genetic bedrock of plant resistance to pathogens? Unless we understand this, we cannot improve plant disease resistance. Plant pathologists have identified different types of resistance. One is non-host resistance, is the resistance in plants belonging to a different taxonomic group. And apparent resistance or a disease escape is mainly due to environment. And there are true resistance. These have been classified into different ways. One is based on symptoms into qualitative and quantitative resistance. And qualitative is no symptoms or hypersensitive response, small speck, as you can see here. And quantitative resistance is degree of disease severity or reduced susceptibility. And also based on pathogen races, they have classified specific resistance and general resistance. Specific resistance is also called vertical resistance, and these are race specific, and other one is general resistance or quantitative resistance or horizontal resistance. Okay. And based on immune response, they have identified pattern triggered immunity, PTI, and effector triggered immunity, ETI. And these two generally considered to induce hypersensitive response. Those that are susceptible or considered as quantitative resistance. My talk will be mainly on this quantitative resistance. Now, the animals in general have two kinds of immune system. One is innate immunity, other one is adaptive immunity. Adaptive immunity is the immune system. It's a very complex in animals. Whereas in plants, there is only innate immunity. So they lead to immune response. And the response will be either qualitative or quantitative resistance. When a pathogen spore or a cell deposits on the cell, it produces elicitor or pathogen-associated molecular patterns. These are recognized by membrane localized pattern recognition receptors or the immune receptors. And they, after recognition, they trigger the downstream genes. And they trigger the MAP kinase and hormones. Okay. The MAP kinase phosphorylates the transcription factors, and the transcription factors bind to the downstream gene promoter and enhance their expression. And these biosynthesize resistant-related protein and resistant-related metabolites. And they, in turn, in the cell, they are antimicrobial and they kill the pathogen. Some of these are very complex and conjugated metabolites, and they are deposited around the infection area, not systemic, but just around the infection area to contain the pathogen to small spot, not a blight. So similarly, some pathogens are highly specialized, like Phytophthora or rust, Puccinia graminis, TTC. They suppress these elicitors they produce. They do not want to wake up the pathogen host, and then they penetrate into the cell wall, produce hostoria. This hostoria produces effectors, and these are recognized by immune receptors or the effector recognition receptors, and they similarly trigger MAP kinase and all these things, leading to resistance. So now, if you look at both these specific pathogens and also general pathogens, or biotrophs or necrotrophs, both can cause hypersensitive response. But generally, pathogens overcome this one and then they attack the plant. This leads to susceptibility or quantitative resistance. So for us, the resistance is, there is no qualitative and quantitative, it's just a shades of gray. It's very difficult to separate. Now, to review this again, pathogens can be biotrophs, hemibiotrophs, or necrotrophs. They produce elicitors and effectors. Both are recognized by receptors that are located in the host membrane. They trigger MAP kinase and phytohormones. This MAP kinase phosphorylates transcription factors and they regulate the downstream genes. These downstream genes produce resistant related proteins and metabolites. They either kill the pathogen or reinforce the cell wall. Now, let us look at the mechanism of disease resistance, mainly due to proteins and metabolic. The mechanism can be apparent, which is generally controlled by environment. For example, a plant with several spikelets very close to each other, contents, they can retain moisture for a longer time, allowing the pathogen to penetrate. If the density is low, then moisture evaporates and the plant may be less susceptible or resistant. That is apparent. And the other one is true resistance, which is controlled by genes. And the true resistance can be constitutive already in the plant or induced following pathogen invasion. Both of these can be structural or biochemical. The structure comes from biochemicals anyway. These biochemicals are metabolites and proteins. Metabolites are biosynthesized from glucose, whereas proteins are coded from genes. So they are not the same. And both of these are produced by genes. Now, in order to select the genes, you have to know the biology of the pathogen and what are the different host layers the pathogen has to go through to get food. And you can reinforce those layers. So for that, we have to know the details of these 4 different groups. Now, constitutive biochemical defense. These are also called phytoanticipants, which are antimicrobial compounds. For example, plants produce the phenolic, flavonoids, fatty acids, and other monomers. These monomers are phytotoxic or not phytotoxic, they're antimicrobial. So, and some of these, like hydroxycinamic, some of these are combined with glycosides and are stored in vacuoles. With simple hydrolysis, this bond can be released and then it will release the monomers. These monomers can be toxic and available to kill the pathogen within seconds. So these monomers again can be from different metabolic pathways. So this is responsible for, in general, why in general plants are resistant to pathogens. These can kill so many different non-specialized pathogens. Now, constitutive structural defense. This gives resistance to entry into the host. This is the host cell. It's a wax layer, and this is the cuticle. It's the epidermal cell. Now, the wax layer, they repel water. Most of the plant pathogens require free water to germinate and penetrate. Bacteria also, they need moisture. If there is no moisture, they die. And there are trichomes in the plant or the hair. They also can, there are several viruses which are transmitted by insects, and they can deter insects. And then the cuticle, thicker the cuticle, and the pathogen cannot penetrate. And the stomata, if the stomata is small or thick, that also reduces. In the cell wall, there are so many components in these cell walls. And to break down each of these components, the pathogen would require different enzymes. If they cannot produce that enzyme, then they cannot penetrate. That's why some pathogens are leaf pathogens, but they cannot attack roots. Those that attack roots may or may not be able to attack the stem. This is the variation because of these things. We have to understand these things. What is the weakness of the pathogen? So this is, for example, this is the middle lamella, and these are the cell walls. And inside are the nucleus and all the things. Now, induced biochemical defense. These are also called phytoalexins. They can be resistant-related proteins or metabolites. Okay. They are generally not present in the plant, or even if they are present, they are in very low abundance. But following pathogen invasion, their abundance increases a lot. And formally, these are known as pathogenesis-related proteins or PR proteins, and there are 14 families of these things. But not all these PR proteins are antimicrobial or resistant-related proteins. Some of the important ones are chitinases, which digest the fungal cell wall, lysosomes, which can digest the bacterial cell wall, and defenses, which can cause pores in pathogen, and thaumatin-like protein, which are antimicrobial. And there are a lot of proteins which conjugate with toxins, plant pathogen-produced toxins, and detoxify. So we can improve any of these genes to improve resistance. Induced biochemical defense. These are phytochemicals, the resistant-related metabolites. They can be monomers or oligomers, like phenylpropanate pathway, flavonoid pathway, fatty acid pathway, terpenoid pathway, the alkaloid pathway. All these pathways produce more than 200,000 metabolites. And as polymers of these, like lignan, wax, and cutin, all these things develop there can be a barrier for pathogen invasion. And also the conjugated metabolites, like 2 or more different pathway biochemicals combine with each other and produce complex biochemicals, metabolites. These are hydroxycinnamic acid amides, suberins, etc. And so many of these phytoalexins, excuse me, they can combine with sugars or glycosides, and they reinforce the cell wall. The induced structural defense, okay, these are, for example, by proteins and metabolites. Hypersensitive response or cell death. Generally, these are due to reactive oxygen species. There are metabolites or proteins. They are produced and they can kill this cell. And papillae or callus formation. Here is a pathogen. It tries to penetrate in, but these callus, they are nothing but 1,3-beta glucans. They are deposited sugar molecules deposited here to prevent the further expansion of the pathogen. But sometimes if these things are thin or delayed and are virulent pathogen, they can break these things and colonize. And others are cell wall reinforcement, like all these things. These are the reinforced cell wall. These are localized but not systemic. This is an induced reinforced cell wall around the infection site, the deposition of hydroxycinnamic acid amides and also flavonoid glycosides. They are deposited on the inner membrane of the cell. This is the vascular bundle. This is the xylem vessel, phloem vessel. This is the resistant pathogen inoculated. This is the resistant mark inoculated and susceptible pathogen inoculated and susceptible mark inoculated. It is very intense here. Similarly, it's very intense here. You must have seen these figures in Agrios plant pathology. So, they block pathogen progress. For example, when you make a small wound
So,
on the tuber,
excuse me, Kushal.
yeah.
So Almaru or somebody can help because your slides are not moving on the YouTube. I can see they are moving in that Zoom, but they are not moving on the YouTube. So many people are asking the question to move the slides.
Okay, move the slide.
Sir, actually, the link is working. If they refresh, then it will move, sir. Not an issue. For me,
Well,
it is moving.
Almaru, I tried to refresh, but they were not working. So I don't know what is the issue.
Now it is working, sir. Like it is
Okay,
working.
good. Sorry, because some people asked me to talk to you about that one.
Oh,
Thank
fine.
you.
Yeah,
Please
no
continue.
problem. Thanks for informing me. If I have to do anything, you please tell me.
Okay, so I think this is fine now. That's what you are saying, Alamuru?
Yes, sir. Fine.
Okay, good, sir. Please, thank you. Continue.
Okay, these are the pathogen progress is blocked by several layers. In potato tuber, if you make a small wound, it develops within a day. It develops a cork layer. Cork layers are nothing but combinations of phenylpropanide and fatty acid metabolites. These are thick and no pathogen can break this down. Similarly, abscission layer. Okay, for example, here, a pathogen is infected here and the cells around it. The host deposits these hydroxycinnamic acid amides or glycosides and all these things. I think so. We have to prove it. And it can contain the pathogen. So instead of a blight, it becomes a leaf spot resistance. Plants also produce here deposition of metabolites. We have to prove it. Tylosis to block the pathogen. This is the xylem vessel. Pathogen generally moves through the xylem vessel and they block it. Also, the plants also produce gums. All these are controlled by genes. So you have whole lot of genes to select. Now, there are whole genome sequences available not only for model plants, but also the crop plants. These, following with the advent of these, several omics tools also have been developed, like transcriptomics, RNA sequencing, metabolomics, and other things. And these have been used to identify the gene functions. So omics to identify resistant genes, both forward and reverse genetics. We have taken, and also others, resistant and susceptible phenotypes, inoculated with pathogen mark, and then they conducted metabolic profiles. These metabolic profiles we identified, and metabolites that are higher in abundance in resistant than in susceptible were considered to be resistant related metabolites. And these metabolites were related to metabolic pathway to identify the genes. These are genes were then silenced based on virus-induced gene silencing and other ways. Then we did we have the edited or the silent and non-silent. We inoculated the pathogen again to see the function of that gene, whether that gene controls resistance or not. Like this, we have proved the functions of several genes. Now, this is the metabolic pathway, okay, and plant biosynthesizes glucose in Calvin cycle, then uses for respiration. This glucose is used in the citric acid cycle to biosynthesize more than 200,000 metabolites. These metabolites can be sugars, terpenes, fatty acids, alkaloids, phenylpropanoids, and flavonoids. And they are produced or biosynthesized by genes or the enzymes. Each of these arrow represents an enzyme. So to produce one enzyme, produce one metabolite, it has to go through several genes. That's why it's a hierarchies of genes are involved, a network of genes are involved. And metabolite production is not controlled by one gene. Okay, so these I will discuss more later. Now, if you look at the overall figure, we have the phenylpropanoids, flavonoids, fatty acids, and alkaloids. These monomers are phytoanthocyanins, and polymers, complex products are phytoalexins. They can be polymers or conjugated, and they are antimicrobial, or they are deposited on the inside the cell membrane to reinforce the cell wall. Okay, if the pathogen is infected here, it is infected around the pathogen infected area. All the cells are reinforced. So the pathogen is contained in the middle, and it starves to death. So I would like to discuss few of the genes that we have already proved in wheat with respect to Fusarium head blight. When 4-CL3 was silent, the biosynthesis of paracameryl-CoA, caffeal-CoA, and ferulyl-CoA were reduced. And these are also called hydroxycinnamic acid thioesters. They combine with amines to produce hydroxycinnamic acid amides. And this gene was localized in QTL2DL. Similarly, we identified another another gene in the QTL Fusarium head blight 1. Here the lactase 4. It biosynthesizes lignin from coniferol alcohol and also sinepyl alcohol into syringel lignin. So when these were silent, the abundance of these were reduced. And the lactase 4 is also regulated by NAC transcription factors. Generally, they may produce very little amount of lignin, but when this is triggered by, induced by pathogen. For example, this is a resistant plant. When you inoculated one spikelet in wheat, it did not move to the next spikelet. But when we silenced this lactase 4, from the inoculated spikelet, it moved to up and down other spikelets. And we call this as Reiki's resistance. I'm sure the same resistance will occur in the stem for so many different pathogens. So similarly, we identified co-comp gene, which converts caffeal-CoA to ferulyl-CoA. Advantage of ferulyl-CoA is it leads to higher level lignin. This lignin, syringel lignin, gives more resistance than these hydroxylignins. So these are some of the genes where we have proved the functions. Similarly, others also have proved the functions. Hydroxycinnamic acid amides are very interesting because these are the amino acids. These amino acids produce amines. These amines combine with hydroxycinnamic acid thioesters. There are 4 different thioesters. These 4 combined together, they produce 12 combinations of hydroxycinnamic acid amides. And there are many more. Okay. We have put only 12 here. I understand there is close to 100 different hydroxycinnamic acid amides. And to make the plant resistant, you don't have to have all these. Even a few can give resistance to a disease. Now, how to enhance multiple disease resistance in cultivar through genome editing? So we do not have to do metabolic profiling of a new cultivar. The R genes, the multiple disease resistant functions of several of these R genes are already known. For a plant to be resistant, it does not have to have all the R genes known in plant kingdom. A few important ones, depending on the pathogen biology, is enough to give high level of resistance. Now, based on molecular biology or molecular markers, we have developed more than 200 cultivars for each major crop. And more we produce, some of the older ones they become extinct. Just because a high yielding crop, well suited to different regions, is susceptible to disease, we throw that crop. Why? And these are the crops maybe we can edit. And the susceptible commercial cultivars also have all these R genes, but only a few are mutated. And if we can edit these mutated ones, we can make that susceptible plant to produce that metabolite, thus the resistance. So this is the overall concept behind genome editing. So unless we know the overall concept, the biology of the pathogen, editing any gene will not give you resistance. Now, in a susceptible cultivar, several genes are polymorphic. Where did this polymorphism come from? Number one is sexual reproduction. We cross male and female to produce hybrids. When we cross, we select for some traits, and for those traits, those genes may be functional. If you did not select some traits, then those genes, for example, for disease resistance, may be mutated. Like this, we inherit in that crop hundreds of mutations. Other polymorphism is the genetic engineering occurs in nature. Bacteria and viruses, they transfer small segments of genes from one plant to another plant. And also in nature, and also artificial rays, and also solar radiation, and also chemical radiation can cause insertions, deletions, and base pair switches. This is the mutation we identified in co-compt gene in Rosette Burbank potato. This is the one I showed you that we edited. Now, what are the candidate mutated genes? The candidate mutated genes for us to do genome editing, they may be regulatory genes. These regulatory genes are immune receptors, MAP kinase, transcription factors, phytohormones, microRNA, and others. There are some transporters and all those things too. They may be RR metabolite biosynthetic genes. They belong to a network of several specific metabolic pathways. We can select some metabolic pathways. Then there are RR proteins, which are also antimicrobial. Now, these are the mutated genes. Where are we going to get the functional genes? The functional genes may be available in several cultivars. Like I told before, A susceptible plant also has several resistant genes, and different cultivars varying susceptibility may vary in some genes are vary in the type of or number of mutations. And also we can obtain this in land races and also germplasm collections. Now, how to identify polymorphic genes in a cultivar for genome editing? This is the beginning of how are we going to do it. Number one option is, if you have lots of money, we can do whole genome sequencing of a cultivar. Second is, we can do RNA sequencing to obtain gene expression and also the SNPs. These SNPs can be grouped into different metabolic pathways. Then, after knowing the resistant related metabolites, then we can say individual genes, we can Sanger sequence to confirm the mutation. And now, we don't have to compare this gene with a resistant plant to see if it is gives resistance or not. We already know so many metabolites are resistance related metabolites. If they can produce that metabolite, then this is a resistant gene. And we can use molecular functions. There are a lot of domains in these genes. If these domains are functional or not, there are several software can identify these things. So we can group the genes into different categories, and some are detrimental, but some may or may not affect. Though there is mutation, they may or may not affect functioning of that gene, or they still produce the protein. Now, gene editing. There are several tools. Today I'm going to discuss only two of them. The number one is CRISPR-Cas9 system with the Gemini virus replicants. Here we use a modular approach. There's a gRNA, Cas9, and donor DNA. We combine these, clone into an agrobacterium, and inoculate to immature embryo or stem. When we produce calli, then they screen the plantlets if the gene is edited or not. Now, how does it work? First of all, this is our gene of interest, and we want to induce a cut here. And this is the Cas9, it's a guide RNA. And the guide RNA will determine or identify, recognize the exact location where you have to cut. And there are several software available to select these things. What is the region you have to select? And they induce, they cause double-stranded breaks. And the plant repairs these things based on the non-homologous end joining. And this leads to silencing of that gene, and that gene may not be able to produce the protein. On the other hand, if you provide a donor with the homology arms to recognize this area perfectly, and this donor will be replaced here. And this is repaired by the plant by homology directed repair system. These are called replacement. So by silencing or replacing, we can edit the genes. Another one is CRISPR-Cas9 based base editors. Here we use cysteine deaminase and adenosine deaminase to switch the CG to TA or AT to GC. In this case, no double-stranded break is needed. Because of this, maybe not only the success rate or the efficiency of genome editing is very high, but also the vector backbone is not generally introduced into the recipient plant. This is one advantage. Now, how to confirm the edited gene sequence? We can, if whatever the segment edited is big, we can do a PCR. If not, we have to do Sanger sequence. And this is the PCR, is the standard, and we can identify based on the fragment. And here is the co-comp gene that we edited in Razer-Burbank potato, and the T was mutated, and now it's converted into G. And these are the chromatogram where all the nucleotides are color-coded. Based on that, we can identify exact location of your edit. If there is any off targets, also we can identify here and eliminate those things. Now, confirmation of resistance. We can inoculate these non-edited and edited plants and produce disease symptom and quantify the disease. This is the non-edited plant, this is the edited. And also in the same area, we can quantify the pathogen biomass based on gene copy number, very specific to that particular pathogen. This is the edited and this is the non-edited. Now, we can also prove the mechanism of resistance. But to do this, do metabolomics. Now, in the Café-CoA produces ferro-CoA, it was very low abundance in Razer-Burbank potato because co-comp gene was mutated. When we edited this gene, the abundance of this increased significantly. But sometimes we may or may not be able to see high abundance of this compound because this compound right away is converted into monolignals, and the monolignals can be used to biosynthesize other higher up metabolites. Even this itself can be converted into combined with fatty acid and others to produce wax and suberin and other things. So metabolic profiling is just a snapshot of a continuous process. So just because we don't find ferro-CoA in high abundance, we should not say, well, this does not do anything to metabolites. We have to look into what are the other metabolites increased following editing of this gene. So it's a trick of the trade you have to know. And now, how many genes should be edited in a cultivar to obtain high level of multiple disease resistance? Now, these are the hierarchies of genes. One hierarchy will produce one metabolite or one protein. If one gene is mutated, and then it will not produce that protein. And if we edit, it will be able to produce that protein. So here it can be deletion or it can be an insertion. Okay. So all these, there are different kinds of polymorphism. Okay, now, depending on the pathogen biology, specific metabolic pathway genes can be focused, and sometimes by editing 2 or 3 genes, we should be able to obtain high level of resistance. But we have to try our luck. But by studying the metabolic pathway, where the individual metabolites are going, we can also predict which gene should be edited. Now, can we commercialize this cultivar? Okay, now several nations have different regulations. U.S.A. is very liberal. Canada and India are following similar approach. European Union, they are not against CRISPR-Cas genome editing, but there is a stricter regulation. Canada regulates based on plants with novel traits. That means no novel genes introduced into the ecosystem, and also no novel proteins and metabolites in the food. You know, so these are regulated. So if we do cisgenic, that means is a, as opposed to transgenic, gene transfer between sexually compatible plants instead of from a bacteria or some other, it is not a novel gene. The gene, what we are doing is already in nature. And it should be transgene free. Because when we are doing genome editing, either Cas9, gRNA, agrobacterium borders, antibiotic selection markers, all these things can be retained in the recipient plant. That has to be screened out. And the food and feed has to be tested. Okay. Though we are doing cisgenic, still like breeders also test all these things. Same way, we also have to conduct this food and feed analysis. Now, how are you going to screen out these transgene DNA? In the case of clonally propagated plants, it has to be screened out in T0 generation because some of these do not produce seeds. So the advantages of base editing is no double standard break needed, and thus less chance of integration of foreign DNA. So, and the efficiency is very high. So it is possible to screen these transgenes in the T0 generation. Okay. Whereas in the case of seed plants, we can do CRISPR-Cas based mutations, mutations and indels. Here, the transgenes can be screened out in T0 generation or in T1 generation. Still, if it is retained, we can cross with the unedited original and select the segregating populations. Here we are not crossing with a completely different plant. It's the same plant bearing only one gene. So it is possible to screen out easily. What are the other challenges in genome editing crops? Now, if you are starting something, always very good to select diploids. Tetraploids and hexaploids, you have more copies of these genes, it gets very complicated. And also, the whole genome sequence should be available for that species at least. Without that, it's impossible. Off targets, generally this is not a problem because if we very cautiously select gRNA, most of the off targets can be avoided. Other one is callus production. Now, some plants are cultivars are recalcitrant. So we have to play with the media composition to improve this. And in-planter methods are available and they need testing. Now, we can register this cultivar. So before registering, we need to do field trials. Extensive field trials are not needed because these cultivars are commercially grown and suited to different regions. All we are doing is improving the disease resistance. So it has to be proved it requires less fungicides to manage or has less disease severity. And we should be able to identify this cultivar because edited and non-edited, they look alike. So we can do a PCR, we can develop a PCR for those genes that we edit. And ownership of these genes are plasmid. If they are patented, we have to obtain permission. Cultivar registration through, well, in Canada, we do it through variety registration office. It depends on the country. So this is a susceptible cultivar. Hopefully we can develop something like this after genome editing. Okay. Well, I take this opportunity to thank all my graduate students and postdocs. And then this is Yogendra, who is in ICRISAT now. This is Gunnaya. Raghavendra is in Horticulture University, Bagalkot. And here is Dhokne, is in Bombay somewhere. And Nancy Soni is in New Delhi. And this is Arun Kumar in North. And Uday Kage is in Australia, and Kare is in U.S. So, and I would like to take this opportunity to thank all the funding agencies. And if you have any questions, I'll be more than happy to answer. Thank you all.
Thank you very much, Dr. Kushalappa. Very nice presentation. I think it was really great to see that you started your presentation from the very beginning, starting from the Big Bang, and then you brought them to that gene editing and then how we can have the better varieties and how they can reach to the farmers. So I think this was really very, very informative, very excellent presentation. And I was seeing that more than 1,000 people right now watching, and many of them, they have given you thumbs up that this is great presentation. So there are several questions, and because of the shortage of time, we may not be able to take all the questions, but I would like to request Dr. Kushalappa for answering a few questions. And one is from Dr. M. Ashraf Bhatt. Question is, and mentioning, we must mostly concentrate on tissue-specific resistance to save energy. Do we have a mechanism in a place where constitutive resistance can be put on hold?
Well, most of the genes we intend to edit are the induced genes. So if there is no pathogen, there is no cost to the yield or whatever. These induced genes are not systemic, and these are local. If one leaf is infected, only around that, not all the cells are modified. The energy is used to contain the pathogen to a small spot. For example, if you take COVID is coming, why so many students are at home? And this is the loss. There is loss anyway. But if you have resistance, there is a minimum loss. I will not say there is no loss at all. More genes we edit, there may be some cost to the yield. But I don't think so. I think it is significant. Hope I answered the question.
Very nice. Thank you. The other question is maybe related to the same from Handling Bong. And this is about that, yes, that's correct. But he thinks that whether this constitutive resistance mechanism may be metabolically costly.
Yeah, constitutive resistance maybe, yes, I agree. Because in thicker the cuticle, most of the energy is used there. So that may be, I agree 100%. That's why we are promoting, or we are doing currently, only induced disease resistance.
Next question is from Sri Monica, and she is asking about induced with the resistance, that what is your view on that one? And do we have some success stories in that area?
Well, induced resistance, we have proved reduced disease susceptibility in so many plants. Or when we silence the gene, the disease increased. For example, in wheat, we silenced one gene, that's lacase 4, and instead of one spikelet getting diseased, the entire spikelet in a spike was diseased. The disease can spread to the entire spikelet. So one gene can give significant amount of resistance, and molecular breeders have identified this in FHB1, which is very stable across different environments, and we think this is the gene. Several other people have shown maybe other genes, but they have not silenced this in a resistant cultivar. We have silenced this in a resistant cultivar. So there is whole lot of proof, and several people have proved by silencing genes, it can increase.
Okay, very good. We may take a few more questions because we know it is very late for you. So one other question is from, I think, Sri Kausalya Palanis, that she would like to know that about some varieties, resistance varieties to control the multiple plant diseases. There are any resistant variety in any crop where you are having the multiple disease resistance? I think this should be the, yeah.
Well, this is the future. Genome editing is the future. Okay. So molecular breeders have already achieved these things, multiple disease resistance.
Yes.
Otherwise, you cannot plant this in the field. So same thing, if they have increased yield, and instead of throwing that cultivar only for susceptibility, we can improve those. That's the overall idea.
No, very good. Thank you. And the other question is from Gopal Singh, and I think this is related to your research. Are you planning to use CRISPR editing to combine HCAA metabolism to develop pathogen resistance?
I did not understand what is the.
So he took this metabolic pathways from your presentation about, and he's asking if or are you planning to use CRISPR editing to combine SCAA metabolism to develop pathogen resistance? He's interested to work in this direction.
Okay, for example, currently my students, one of my students is editing the co-comp gene in a commercial cultivar. Rajiv Burbank potato is grown in 40% of the land area in Canada and US. In that, co-comp gene is mutated. And when he edited, it increased resistance significantly. Now, but that plant has vector DNA. So what we are doing now is in potato, we are doing base editing to screen out vector.
Okay.
So whereas we are also working on wheat and barley, there we are doing mainly CRISPR-Cas9 with Gemini virus vector. We have selected a few genes.
Okay. And this is one is technical question from Kahelon, and his question is how to screen tDNA free plants, how we can confirm tDNA integration events effectively, what if small tDNA fragments cannot be amplified by simple PCR?
Yeah, there are deep sequencing, amplicon sequencing, and now there are so many tools are available. These are, but mostly the small DNA, small fragments get eliminated, especially in a segregating population, the T1 generation or T2 generation itself. These are not very stable. They get eliminated. If at all the Cas9, this is the major one, the Cas9 is eliminated, then rest is not that problematic. And also we have to be careful in using any antibiotic selection markers.
Very good. So
Yeah.
I think, thank you very much for answering several of these questions. And I would like to encourage colleagues who have asked the question, and they can send the request, or if they have some specific question, to Dr. Kushalappa by email. And they are most welcome to keep on viewing this presentation again on the YouTube to understand further. But for me, this was very, very comprehensive and excellent presentation. So I have been heavily benefited. Thank you very much, Dr. Kushalappa, for great presentation. And also thanks to Plantgenomia for organizing this thing. And I think I hand over back this session now to Plantgenomia, and they will continue now for the pending activities. Thank you once again.
Thank you.
Well, that was extensively informative. Thank you very much, Dr. Rajiv Varshney, and our speaker, Dr. Kushalappa, for such a flawless conduction of events and for illuminating the subject to this extent. Thank you very much.
Pleasure.
For the next event, I would like to call upon Dr. Sujaya Sri, Chief Operating Officer of Plantgenomia from Indian Agricultural Research Institute, New Delhi, for carrying out the interview round.
Yeah, well, success is different for everyone, but it is always interesting to learn how people arrived where they are. So, today we have an honor of having two outstanding, successful scientist personalities with us. That is Dr. Kushalappa sir and Dr. Rajiv Varshney sir. Sir, we are going to have a really interesting talk with them. I would like to ask few questions. Which would definitely gonna inspire our audience, sir. So I will begin with Dr. Kushalappa sir, and then I will move to Dr. Rajiv. Kushalappa sir, we would like to know what has helped you to be successful, or sort of challenges you face to attain this position, sir.
Well, the challenges are many. Okay. Okay. So now to start with, I was an epidemiologist, South Pole, and my genetics and molecular biology is about 50 years old,
Okay.
or 60 maybe, 50.
Okay.
Okay. So I don't know genomics. I don't know genetics. I don't know molecular biology. So I was working on epidemiology. Then I moved to post-harvest pathology. And we used to quantify metabolites in the headspace to detect the disease. With the advent of whole genome sequencing, other omics appeared. There was a conference in Boston on metabolomics. I attended. Oh yeah, we can do this. So then I subsequently attended 3 or 4 or 5 metabolomics workshops. Slowly we picked it up. Then we were the first one to prove large-scale metabolic profiling in resistant and susceptible plants and show metabolites are very, very important in disease resistance. Then what do you do with metabolites? Can we do them as a marker? Impossible. These are controlled by genes. So I'm not a geneticist. There are so many labs with all kinds of expertise, but there is no communication. One person plays Beethoven, other one is, sitar, other one is some other music. Very difficult for them to understand each other. So what I did was, I went to Bangalore, visited Ramanjani Gowda, and gave a talk there. And hired a couple of people there. I told them, you guys teach me genetics, I will teach you plant pathology and molecular biology. Learning is never ending. I read every day like graduate students. I am a graduate student all my life. So Those are the challenges. Unless my students have lots of ideas. I don't know, maybe because of that, even me also, when I graduated in Bangalore, came to Florida, I could memorize so many things. But when we went to lab, my other classmates, they used to finish the lab in 1 hour. I was still struggling for 3 hours, not done.
Okay.
We have two issues you students should know. Number one is you are not encouraged to think. Okay. Professor knows everything. No. Here is the example. I don't know genetics. So student knows he is my professor. I told you are my professor. I'm a professor here. Then they say, okay, you have not answered the question. And I ask again, coach, I told you last week. I said, well, you are not a good professor. You did not teach me in the way I understand. It is your
Okay.
duty. It is your duty to explain to me in such a way that I understand. Not that I understood the whole thing, but I have the logic. So challenge is with logic, you can make decisions.
Okay, sir. It's inspiring words from your side, sir. And
Thank
moving to the next question, sir, I have one question. As most of our audience are postgraduate students and PhD students, so we would like to hear from you the inspiring messages for the research career, sir.
well, well, there are, I don't know if we have too many graduate students. Now, every day, every other day, I receive an email for a postdoctoral position, for a PhD position. So there are so many people willing to pursue their studies. You have to pursue your passion. Just because one university says no, don't give up. Because each professor, each university, each province or a state, they have different funding systems.
Okay.
Sometimes we get funding, sometimes we don't. And so now all the funding is related to industry. Government does not give funds unless industry contributes 50% or 25%. Now we don't have, we have lots of projects, but no industry is coming forward.
Okay.
If you don't have funding, we cannot hire you because the student needs about 15,000 minimum to maintain, and another 15 to 18,000 is the fee. So wherever you apply nowadays, the websites are well developed. Visit the websites of the university. Write down how much expense you have, how
Okay.
much money you have. If you are a Bill Gates daughter or a son, no problem. You can come. I don't take self-funded students unless they are really super rich.
Okay.
Okay, so different universities, different states have different uh, funding. So you have to try to develop a good CV, explain your skills. We look for skills and
Okay.
do not hide information. The moment you hide information, we reject that application. Okay.
Okay.
Mainly we consider your undergraduate, the overall grade point average.
Okay.
We have high grade point average, we consider. And
Okay.
often we don't consider recommendations and all because everybody gives good recommendation. But still that is needed. But these are the important. You have to sell yourself. Consult people how to make a good CV.
Okay.
Yeah.
Thank you, sir. Thank you for your guidance. And sir, my next question is, sir, you work as a scientist. Your work as a scientist is well recognized globally. So we would like to get insight into your vision in emerging technologies in genomics and CRISPR, sir.
Well, the world has invested billions of dollars in sequencing and all these things. And we did not know so far We had only how does all the developments occur. When microscope was invented, they identified, okay, there are plant pathogens, and the pathogens are not coming from rain. Like that, the development in science is mainly due to technology. And today we have an excellent technology, that is genomic information.
Okay.
And this will stay for another 100 years. These are the fundamentals. More you know, better it is. You have to know omics. If you know just genomics, not enough. You have to have a good molecular biology background, some bioinformatics background, and whatever the trait you are improving, that background. Some students ask me, can I, does it matter which crop I work? It doesn't matter. Do you know the tools? I mean, the skills.
Okay.
Skills are the ones we are using to hire people, not only for students, but also maybe Rajiv can tell what criteria he considers to hire people.
Okay, sir. It's a great words from you. And sir, one more question, last question I have for you, sir. Like, sir, as you are one of the renowned top scientists cited in the world, we would like to know your opinion and suggestions in scientific writing and publications, sir.
Well, publish or perish. That is the dialogue everywhere. So you have to show. You send me a CV, I don't trust you. Somebody writes you a recommendation, I don't trust you. When you publish a paper, then I know, I read those things. I know, okay, you can do this, you can do that, and all those things. Publications are very, very important. Okay, and
Okay.
also sometimes we call up and ask, who wrote that paper?
Okay.
For example, one of my PhD students was interviewed for a job in some country, and that person called me and asked, this person has published so many papers, and who wrote that paper? I told, it's a rule in my lab. The student writes the first draft. Sometimes those drafts may go for 20, 30 times, editing back and forth. But still, the student writes the full draft first. I do not look at the paper until he writes that. So all my students write that paper. Then I ask him, why do you ask that question? I have a better candidate, but his boss wrote all the paper. But he's a very good researcher. So now improve your writing skills. Very important.
Okay, sir. Thank you for your. We are grateful for your suggestions and we'll take them to our heart. And thank you from the team Plantgenomia, sir.
Thank you.
Yes, sir. So moving to Dr. Rajiv, sir. Sir, first of all, we are congratulating from Team Plantgenomia for listed as 123 in ranking as one of the highly cited researcher in 2020 in plant biology and botany, sir. So congratulations on your well-deserved success, sir.
Thank
Yeah.
you very much. Thanks very much, Sujasri, and all your colleagues. Thank you.
Yeah. Sir, moving to the questions we have. So we would like to hear from you the same question, the steps how your life has shaped to be a successful personality.
Thank you very much for this question. So I don't consider, well, I'm satisfied what I am having right now, but I think that this is my suggestion to all the colleagues who are attending this mm, seminar. So I did not have any formula, but one thing which I always remembered, and I think thanks to my mother, to whom I used to respect a lot, so I am very heavily impressed by her. She always used to tell that just keep on doing hard work. And as in Indian mythology or religion, we say like Gita, that you should not desire for the result will come automatically, and only hard work is in our hand. And that's what I have been trying to do since my school time. And the other good thing, which I think that I have been very privileged to have wonderful mentors, teachers, and nowadays a wonderful team of my colleagues, my PhDs, postdocs, colleagues, and collaborators. So I think I have been, rather, I will say not that in the area of successful, but I will say that I have been really privileged to have wonderful team, wonderful colleagues, and my well-wishers and mentors around me. So they really saved my career, and I'm really grateful to all of them.
Yeah, thank you, sir. Thank you for your inspirational words. And my next question is, sir, based on the email request we obtained, we got to know that genuine interest of many of the dedicated and passionate students in pursuing a program or a course abroad. So, sir, what sort of guidance would you suggest them in getting into?
This is very good point, and as Dr. Kushalappa mentioned, so I think this is good that nowadays we are having large number of people interested in science, and this is really good for the scientific community.
Okay.
And what they need to do, that they really need to put their details in the CV in very nice and reflective manner, and they should not be basically have very crisp summary, not telling everything they know. And if you will write these kind of things, then many times the professor who receive these CVs, they think, oh, maybe this is not correct, this is not correct. So these are the issues. Second is that especially for those students who are doing PhD, they should not wait for their publication. After that, they think that, well, let me complete my PhD and then I will start to publish the paper. And that I learned from my professor, Professor P.K. Gupta, when I was doing my PhD here in India, that when you are doing your research, so during the course of your PhD, you should publish the paper, because in your CV, when you go for postdoc application, or when you go for scientific or job or so, it's good to write that, well, I have done this sequencing, I have done this gene editing, I have developed this variety. That's fine. But these things should be in the form of peer-reviewed papers. So I think these papers are really important. So if you are a young person, I think I will put a lot of emphasis for you guys to prepare these good publication. Don't go for the predatory journals, and go always in the good journals, and try to put these things in those. And by doing this thing, your PhD, when you defend your PhD, when you write your PhD thesis, this will also be very easier. So in my opinion, again, my suggestion will be to focus more on really good research outputs and keep on publishing them when you are working for your PhD degree.
Thank you, sir. It's a very thoughtful idea, sir. Yeah, sir. Moving to the next question. Sir, we would like to know your personal opinion on research collaborations with different organizations in terms of sharing the knowledge, skill, and techniques to get mutually benefited each other.
So yeah, you have asked very good question, and this is the requirement, very much requirement in the present time.
Okay.
And many international organizations, many developed world, these things are happening. Sometimes in some organizations, in some countries, these things do not happen. And I think, in my opinion, nowadays things have become multidisciplinary. And if you would like to address any challenge, you should not work in silo. We need to work in collaboration. But what is happening, that many, or not many, some of us are a bit afraid that if I will collaborate, then that person will take over my credit, or why I should share my knowledge, etc. Our laboratory has been doing large number of collaboration around the world. In some collaborations, we were not successful. We have burnt our fingers. But at the same time, by having these kind of bitter experiences also, we did not stop to collaborate. We continue to collaborate. So in collaboration, I think mutual respect is very, very important. Due credit sharing is very, very important. And all of us should learn, or all of us should work in this direction. And I always have this proverb I tell even my PhD student, my colleagues here in ICRISAT, and I tell in the lab that, well, that we should not have collaboration within lab only, within the institute and with outside world as well, because we alone cannot do everything, but together we can do something. So I think that we should always remember, and we should have the multidisciplinary collaboration. If you will do like that one, then we can make much more better impact. you can have very high quality publications as well, and you can have very high quality outputs, which will be very application oriented to contribute to the farming community in developing countries. So that, I think that will be my take. So we need to continue to collaborate and always think from the broader picture, not that smaller pieces of the puzzles, but right now the whole puzzle, that how we can make the whole puzzle. So this is very important.
Great vision, sir. We appreciate it, sir. And sir, one more question, similar to the question I previously asked. Sir, the budding researchers are facing challenges in their researches and publications, sir. So, how to tackle such kind of problems for the new researchers?
So this is very important point. And whenever we have these young students, early career researchers in our lab, and I keep on interacting with them in large number, and I always feel very happy and excited. So in my, and I, okay, yeah, so this answer will be a little bit longer, but what I wanted to tell, I think this is also a responsibility of the senior scientist to help early career researchers. And I think this is our obligation. And as we say that science always grows like that one, and is this at the standing on that shoulder of the giants. So I think the first is that senior scientists, they should find time to mentor the early career researchers, not only from their lab, but even from the other side. And I personally feel very excited when some early career researcher or people, they say, Rajiv, we learned this thing from you. Rajiv, can you please do that one? I try to answer all the emails when I receive from early career researcher, because I know that I was also in the similar position a few years back, and we need to encourage these people. So this is the point number one, that we need to help these colleagues. Second is, these people, early career researcher, they should not go in that shortcut scenario that, well, I need to publish the paper, so let's copy paste, or let's do some wrong thing, unethical thing for completing of our experiment. No, we should be very ethical in science. We should not play with the data. We should be really work following international standards, high quality standards, because once you publish the paper, you cannot track it back. You cannot retract, which will be very shame if something happens like that one. So you should make sure that all the data, all the analysis going in that paper, which you are putting, is really of high quality. Third is how to write a nice paper. What happens that many times nowadays people are not reading many papers. I was very happy to see what Dr. Kushalappa told, that, well, he's reading now. And I know that from my professor, Professor P.K. Gupta, who is going to be 84, in next 1 or 2 days will be completing 84th birthday. And I'm so much impressed with him that he doesn't have right now teaching responsibility, etc. But even today, he keeps on reading the paper. And many times he calls me and says, Rajiv, did you read that paper? I say, well, sorry, sir, I have not yet read. No, you should read. So what I'm trying to say, unless you read many more papers, unless you understand those papers, and you cannot write a good paper. So first is, nowadays what is happening? Many times, and I tell this thing to my students, they download the paper on their computer. When they download, then they think they have read the paper. Downloading is not reading the paper. You need to read the paper. You need to understand how these things are written also. You should, and after that, you will read the paper, bring 4, 5, 6, 10 different papers together and try to see that now I should not copy and style of any single paper, but based on these things, I should come up with my own ideas that how I can present this story in the nice manner. Many colleagues, they ask me, Rajiv, you have been very successful to put so many papers in the nature journals and the top class journal. What is the thing? Some people say, hey, we also sequence the genome. Our genomes are not published in those papers. How your genomes are being published in those papers, or how you are published? I'm having a lot of colleagues from China, from many developing countries, and many other places that they always ask this question. The thing is, this is not important what data you have generated. The most important for publishing the work is that how good you have been in analyzing the data, how good you have been in interpretation of the data, how good you have been to present the story. And just sequencing something, just genotyping something, just generating something, and you say, well, I have done it, but I was not successful to publish the paper, and we start to blame the editors. This is not the good thing. Rather, read more paper, based on these papers, come up with your own style. So this is the thing. Other thing is, and I am still learning writing papers, and I am also being, many of our papers are also being rejected every day. So this is not that We are always successful, and what you see, you always see the success stories. In our laboratories also, we have many failures as well in terms of the experiment, in terms of the papers. So that's not a problem. We should not be feeling discouraged with this rejection thing. And I always tell, if you have written a paper, this will be published. Maybe that your paper was not fitting in the theme of that journal or something. So again, you need to come up with your own style, and you need to, and then if you will do these kind of things, I think you will be very successful to put these papers. Many times, being coming from India, I know that we have this habit to write very, very long sentences. When I checked the PhD thesis, one sentence is completed in one paragraph. And unless you can understand your own sentences, you should not think that, well, you need to write this thing in the paper, so you should have the smaller sentences. There are many things, and nowadays you can find these tips on the internet as well, that how to write and impressive papers. I also gave the talk once or twice, or I think several places, including in Australia or so, that nowadays this is very important for early career researchers, how to write the nice paper, because this is very important. So very good. Thank you very much, Sujasri, for asking these kind of questions.
Encouraging, sir. It's really encouraging for the new researchers. Sir, lastly, I would like to ask you one more question. What kind of the message you wanted to convey to the younger generation to build them as a passionate science researcher?
Very, very good. So I think many things were said by Dr. Kushalappa, and also I mentioned some of these things. So what we need to do, we need to be really very committed for something. What is happening that this new generation, and I also have seen this thing, and so all of us would like to achieve something in very short time frame. And and if we are not reaching that goal, we think it's good to change the goal post.
Okay.
My feeling is that you should have the determination to achieve your goals, pursue your goals, keep on working hard in that direction. And if you are not successful, this doesn't mean that your hard work has gone waste. Rather, you gain some experience. At least you now know that how to write a paper, how to present a paper, how to present your findings, how to present your results. So these kind of things will be very useful for the future researchers. And I think
Somebody should telephone and tell.
Maybe the internet issue is there, like he'll be joining in 1 minute. Yes, he is about to join in 1 minute. Like we'll be waiting for 1 minute.
Sure.
We are sorry that Dr. Rajiv Varshney is having some power fluctuations. So meanwhile, we'll be continuing the other part. If he joins, and we can ask this last question to him. I
Yeah,
hope it is okay, Suji. Yeah?
yeah, it's okay. And that was my last questions to sir actually. And Kushalappa sir, your comments on our organization or anything to talk about Plantgenomia, sir, you can.
Yeah, I really congratulate you guys, young minds. Yeah,
Thank you, sir.
you are using your creativity is very very important. Everybody, our conventional, okay, do this, continue in the studies. No, you have to come up with new ideas, and that will help you and also your colleagues. And this will give you also kind of international exposure. That's also good. Invite other people, and similarly, what is useful for your uh, group of people who have registered. You
Okay,
have to
sir.
ask them what interests them, what is the national issue. Okay, so then you can invite some people in that to talk about it.
Thank you, sir. Thank you for this valuable word.
Sujya, a moment. Like we are having Dr. Rajiv Varshney back with us. So the last question, what we missed from him, you can ask now.
Yes, sir. Sir, I was asking like the inspiring message you can give to convey to the younger generation
Yeah,
who are very passionate about their science researches, sir.
sorry for this net issues, but I'm back again. So I think I already mentioned a few points. So for instance, that we need to have our goals,
Okay.
keep on working hard in this direction. And if we are not achieving success, this doesn't mean that that you need to give up. Maybe you learned some experience, at least that you know now that what you should not be doing, that you will not be achieving that one. So in my opinion, that continue to do hard work, continue to have your commitment, and don't go in this basically shortcut road, because these are not really good direction to achieve your goal. And I think that when we have some goal that I need to contribute in this direction, you will be very successful. Sometimes you may be having failure, should not be discouraged. I think all the successful people, what we see around the world, we should not think that they always had the successes at each and every step. All of them and all the senior people who are successful scientists, when I talk to them, I have realized that how much difficult time they had at different time points. So we should not be worried about that one, that, oh God, why this happened with me or so. So these are some of my suggestions. So don't worry about these things. Keep on working hard. And that's what I would like to say. Thank you very much once again, Sujasri.
Yeah, thank you for enlightening us, sir. And sir, it was very fascinating talking with both of you, sir. And we feel grateful to listen from both of you. And thanking really for sharing your knowledge, experience, and valuable insights here. So it was really exciting insights into the world of science and motivation. Thank you, both of you.
Thank you.
Thank you.
Truly an amazing interaction to have waited upon. Thank you, Dr. Sujashree, for executing the interview, which most certainly was quite intriguing. Now, wasn't that a mind blowing session that we had? So, thanks a lot. You guys have been a brilliant audience. And I would also like to acknowledge the efforts put in by the core subject and the core support teams, the chief guest, and the speaker, of course, Dr. Rupali, Dr. Sujyashree, a media partner, Just Agriculture magazine, and all my fellow workers who contributed to make this event a success. Reaching the end of this, I would like to clarify the process of attaining the certification for the same. A feedback and certification link has been shared in the chat box and the YouTube comment box as well. You're all kindly requested to go on to the same, fill it up, register yourselves, and thereafter, within a slot of 48 hours, your certificates shall be shared accordingly. And if there is some error and you do not receive your certificates after 2 days' time, kindly visit our official website www.plantgenomia.com. Please note that the link is valid only for 2 hours. For further clarifications, kindly visit and subscribe to our YouTube channel to get the detailed directions for certificate collection. With this, I would like to deliver the vote of thanks for this event to all dignitaries assembled here. To begin with, I would like to thank our chief guest who honored this function with his overpowering knowledge and inspirational thoughts. Also, I would like to thank our speaker today, Dr. A.C. Kushalappa, for having embraced us with all his fair insight on this topic. Our media partner, Just Agriculture Magazine, the Chief Operating Officer, Sujya Sri ma'am, and our co-host today, Dr. Rupali. The participants, course support and subject specialist team for the valuable support and patience exhibited by them. I finally take immense pleasure and this opportunity to conclude this meeting with the righteous success that it has reached. Thank you everyone for joining us today and contributing the biggest piece to this event's success. Thank you.
Thank you very much.
Thank
Thank
you.
you, everybody.
Thank you very much, sir. Thank you once again.
Very nice. Thank
Good
you.
luck.
Thank you very much,
Yes, sir.
Dr.
Thank you very
Kushalappa,
much.
once again.
Thank you, sir.
Thanks, everybody. Plantgenomia, guys, keep it up. You are doing great job.
Thank you
Yeah.
very much,
Thank you,
sir.
sir.
Thank you.
Keep it up.
Thank you. Thank you, sir.